Well, hello everyone, and welcome to today's webinar entitled The Future of Foliar Disease Identification, Quantification, and Impact Determination. I'm Chris Boomsma, Director of Education here at the American Society of Agronomy and Soil Science Society of America. It's my privilege to once again be today's webinar moderator. And with that, we're going to kick off today's event. First of all, I want to thank our sponsor. Today's sponsor is the United Soybean Board, USB. They have been a sponsor in the past, continue to be so now, and that's fantastic because it is bringing you quality educational material at no cost. So the ability to pick up a free CEU and learn a whole lot is very much appreciated. So we thank you, United Soybean Board. Now, speaking of the United Soybean Board, I want to take just a moment to promote something that they've developed, and that is the Tech Toolshed program, which is a soy checkoff resource to help soybean farmers maximize their current technology, integrate new technology, and manage available data. USB realizes that on-farm technology and data management services help farmers make better decisions. They boost efficiency, decrease inputs, increase yield, and help them be more sustainable. But there are few unbiased sources of information on technology. So for farmers, it's challenging to navigate all that. The Tech Toolshed helps soybean farmers investigate and adopt ag technology appropriate for their farm and incremental steps. It's a launchpad for helping soybean farmers implement technology and data to improve best management practices and their bottom line. So you can see on your screen right now, visit unitedsoybean.org/Tech Toolshed or follow it, that is the Tech Toolshed, on Twitter. So thank you, USB, and please folks, go check out the Tech Toolshed. Okay, now, as we always do with our webinars, just a few key notes. First of all, please contact Lacey Edwardson. Note that is a new contact. We've had a staff change here. Michelle Lovejoy, who's been doing this for so long, has moved into a different position here at our organization. Lacey Edwardson is now your contact for customer service needs with our webinars. Please note her email address, phone number on the screen. At this time, if you have any issues, please reach out to her first. Please note that this webinar is worth one CEU in pest management or professional meetings, depending on the certification you have. Next, as always, we're going to have a question and answer period at the end of the webinar. Type your questions in the questions portal and go to webinar. Submit them to me. I will take a look at them, order them, get them ready, and then pose them to our presenter at the end of the webinar. Note that this training session is being recorded. You will have access to both the recording and the slides 1 week or hopefully less after completion of this presentation. Also, be on the lookout for future webinars. We have one later this week, and then we're going to take a little bit of a break as we wrap up the growing season. We go into fall, but come late October, November, December, January, February, it's going to be hard and heavy with webinars. So please come back for some webinars. There will be some free ones. There will be also some great at-cost ones. And there's always the online subscription, which you can purchase and get access to them all. So with that, on to our presenter. Today's presenter is Xavier Phillips. He's a plant pathology specialist at Syngenta and also a PhD student. He holds a master's degree in plant pathology and microbiology from Iowa State University. He is currently completing his PhD in the Department of Plant Path and Microbiology. His graduate work was primarily consisted of field experiments involving fungicides and their impact on soybean diseases and disorders. He's experienced with remote sensing, image analysis, unmanned aerial vehicles or drones or UAVs, however you call them, and global positioning systems or GPS technologies. He's also an avid gardener. So it's not often that we actually have a PhD presenting, a PhD student that is, excuse me. But he was highly recommended. He's doing some cutting edge work in this space, and he's a great presenter. So we look forward to hearing from you today, Xavier. We do appreciate you being here. And with that, I'm going to turn over the presentation privileges to you. When you are ready, go ahead, show your screen, and we look forward to your presentation.
Okay, thank you very much. Can you see my screen all right?
I can. It looks fantastic. Take it away.
So thank you, everyone. And first things first, because I believe this is the most important and shouldn't be reserved for the end, but the acknowledgement. So Chris, thank you very much for getting me set up and taking care of everything. Darren is my advisor, and he gave me the heads up about this opportunity to be here with you. And thank you for everybody for showing up. and I especially hope everyone's doing all right after the storms that we had yesterday in Iowa. That was not fun. So some of my funding came from USDA Hatch as well as USB, so I thank those two. And I thank USB in general for putting this on. So importantly that we don't forget that all of these steps that we take towards progress now are because of the strides that those before us have done. So let's not forget that. And moving on to the outline of my presentation. So I'll talk about UAVs. I worked with them throughout some of my research, and plant diseases, of course. I'm a plant pathologist. We'll look into identifying, rating, and their impact they have on yield. Then we'll move into teaching machines, and I hope that interests you because it's something that's really moving quickly, especially in precision agriculture. We'll talk about this parameter called RUE, that stands for radiation use efficiency, also known as light use efficiency. And then we'll look at bringing all these things together, UAVs and algorithms and maybe some other forms of remote sensing to move towards the absolute estimation of RUE. So UAVs first. They've gained a great deal of popularity. They're fun to fly, but they also can do a great deal for us, especially with collecting data. And let's look into that. So UAVs are not only for giant fields and looking over huge acreages, and they're great for doing that because they can give us a bird's eye view of what's going on. The average farm is around 450 acres, and getting an idea of what's going on in the middle is tough. But we can also use these for research. And researchers like myself, we look at a small plot scale. So these UAVs also have the capability of providing ultra high spatial and temporal resolutions. And that's what you need on a small plot scale. So here's an example of maybe a big farm or a field. So this is, it's not an image, it's an orthomosaic. It's a bunch of images stitched together. And so we can get awfully high resolutions from that process. And we can get an idea of what's going on, and we can use this for scouting. So at least in precision agriculture, we can get an idea of where some things are going on in the field. So then we can go out and verify and check on those boots on the ground. So, but researchers need to get a little closer. And as I said before, we're able to do that with UAVs. So here's an image of a UAV flying about 30 feet above soybean crop, above this plot. So you can see, if we all know, when you zoom in close enough, you start seeing all these pixels, and those are, you have to zoom in a lot. So that 1x1 sheet of paper there on the left image, that's a 9x11 piece of paper. So you can see that we're getting very close. And I mentioned before, pixels are the limit. That's where all of our data is held. Those are like the vessels. It's the smallest unit of this image. And contrary to what some of those popular crime investigation shows might have you think when they say enhance and they somehow zoom in past the resolution and find some new data, that doesn't happen. You can't go any further than what you see. This is the limit. This is where the data is. There's nothing beyond this. And within these, there are layers and layers of data. And so UAVs are fun, but those are the platform So when I say platform, they carry what's capturing this data. And what's capturing the data are these sensors. And so we have a standard sensor on the UAV. And then if you look again, there's another one mounted in the back collecting different bands of imagery. So all of these are data. So when I say bands, we look at a common RGB image and we see something pretty, but it's not just one layer, it's multiple layers stacked up in a certain order so that it displays something, and we can rearrange these layers to display some other things of interest. And there are many other layers, if you will, or bands of imagery that we can collect depending on the sensor that you have attached to your UAV. And so here's just some. So those RGB, those were just in the visible spectrum, about 400 to 700 nanometers, but there's much more that we can capture with different sorts of sensors. Some popular ones now are thermal and near red, but very common ones are NIR, which is near infrared, which is used for some popular indices such as NDVI. And so image indices is just a combination of bands. And I'm sure you'll hear this. It's common, and they emphasize a specific phenomenon. And then they also mitigate unwanted noise. So we're able to use these indices to pull out things that we may not see just by looking at a plain RGB image, as we were before. And so NDVI is a very popular one using NIR and the red band. And then the EXG, which stands for the excess green index, is a popular index used for discriminating between green leaf area and background. So that image there from Varela et al. 2018 is actually looking at corn seedlings and doing automatic stand counts. So I think we've all done stand counts, or likely most of us have done stand counts, and those are not enjoyable at all. And this isn't new, it's been going on for a while, doing automatic stand counts from UAV imagery. Some use NDVI to identify plants, and here's an example of EXG doing it. But we can do that across not only research fields, but also perhaps farmers' fields to get an idea of replant decisions as well. So going back to your RGB, your layer of red, green, and blue, but there are those different layers as I spoke of before, and perhaps those layers will give you an idea of something else going on in your field. And so then you may want to investigate if you're a farmer, or if you're a researcher, perhaps you use this different data layer or this combination of bands as an idea as to where some of your error occurred during your experiment and accounting for that. So that's important. And so again, there are many, many indices. You could make up your own indice. It's just a combination of bands. And so imagine that this is a big, big field, and our farmer's using his UAV to get some pictures, get an idea of what's going on. This is closer to the end of the season. Well, it's not early in the season, but we get up in the air and he takes some images and he sees, oh, goodness gracious, in the middle of my field, I have something going on. I have no idea what that is. I should probably check that out. As I don't know how likely it is that maybe you or someone who was scouting your fields might have seen that during the season, as it's in 450 acres. At least that's the average size. Some are much bigger, some are much smaller, but that's in the middle there. So that might be tough to spot just from the ground. Then we see some other areas. Oh, we know what that is. That's at the edge of the field. I know what happened there. That's a low spot. But then I see some of these other spots I identify, we should check those out, get someone out there to look. Oh my goodness, and then we see something like this. I don't know what that is. Lots of spots on my plant. There's a lot of them. How much is there? I don't know. I don't know what it is. We need some help. What do I do? What's it mean for my yield? And then if you think it was corn that you planted, then you might definitely have some issues. And that before, that was frog eye leaf spot on soybean. So that's a disease, a fungal disease, and diseases in general do a lot of damage and cause a lot of loss. So at least this recent paper just put out this year looked at 2 decades from 1996 to 2016, and they saw that annually there were losses due to soybean diseases that reached nearly over $4.5 billion. And they saw also that most of this loss was up in the northern states, and that makes sense. There are more in the north. Soybeans are grown more, so there's more beans. When you have more of something, you can lose more of it too. And so that was between those 2 decades. And so as we go into figuring out what it was, how much is there, and what do we do? Well, there's this thing that is called inter and intra rater, these things called inter and intra rater variability. So researchers are familiar, certainly, when we walk out into a field, or pathologists, we walk into a field and we need to rate the disease that's there. And rating is difficult, and it's a big issue because it's done by people, and people are the biggest source of error, probably no matter what we talk about. And so we're rating this, and there's variability. So between you and I, we go out into the field and we see this leaf, and this is frog eye on soybean, and we say, you say, that's about 5, that's 6% of the total leaf area that is taken up by lesions of this disease. And then I go, I think that's 30%. So there's a big difference between your rating and my rating. That's inter-rater variability. And then if it's just me, and I come to this leaf and I said, ooh, that's 30%. And then I go into another part of the field and I see a similar leaf with similar amount of severity, true severity, and then I go, wait, I think this leaf is 10%. But now you and I both know that that's not the actual severity, and it's much lower, but the 2 ratings I just gave for the same image that I just flipped around is very different, and that's intra-rater variability. And reducing this is important, but it's hard to do, not only for trained pathologists, but for anyone in general. And it's frustrating. And there are tools that we can use to help us calibrate ourselves before going into the field. And one of these is from the Crop Protection Network. And this is a disease severity and defoliation training tool that they have. So you can go onto this website, go onto their website and do this training, and they have multiple diseases for corn and soy, and you can click on one. I clicked on soybean here, I'm showing you, and you can choose a disease, and then you can see the disease show up on the leaf, the representation of it. And then also you can continue, and you have a tool which you can slide across, so you can see examples of, here's 20%, and you can slide it further to see what 50, 70, or lower to see what it looks like. And then you can take a test to train yourself, and you'll be provided with images, and you rate those images, and then you get a score at the end, and it shows you how you did compared to the actual severity. And so this is very helpful, and I encourage you to look into it. And we'll take a little turn, and I'll show you how it all fits together. So here's a paper that was published in 2017, and it's titled "A Deep Learning Framework to Discern and Count Microscopic Nematode Eggs." So if you know any pathologists, maybe during graduate school, I'm sure they said when you asked them, "What are you doing today?" Counting eggs. It didn't sound fun. I never counted eggs. But this was a seminar given in 2013 before they published this paper, and this gentleman here gave that seminar. And one of the things he said was, at the end, was, if you can identify something, then we can teach or train a computer to identify as well. And that, I thought, that's amazing. You can, if I can pick out something, then we can train a computer to do that also. That's amazing. And just to show you what they've, a quick work through of what they were doing. So for the nematode part, you collect your sample and then you process your sample and you take these images. And so let's focus on that top part right now. So commonly, if you have 3 experts, as you can see, and we're counting those eggs on those plates, that first expert there is doing very well. That little call out right there, he's full energy. So from the start to the end, he does not have much variability between what was actually the eggs that were actually on those plates to what he counted, and he did very well. And then as we move to the expert number 2, he must have skipped breakfast, or he was very hungry. It's probably close to 12 or maybe 11:30, and he's just ready to get through these plates. So he doesn't do that well. And then the next expert, well, he had breakfast, so he's doing okay, but not as great as the first one. So there's a lot of error between these experts. And we can reduce that, and they show that in this paper. And so this is where we come across deep learning models. I thought this was very interesting. And you can apply this to not only nematode eggs, but if you can identify in an image foliar diseases, wow, I'm sure that's something many more people would be interested in, and it is, and that's moving forward. So just some things of what they found, they saw that they could maintain human level accuracy counting these eggs, but reduce the error that comes with humans. So that's quite amazing. And they could do this in images that were very messy, filled with debris, and it still worked out. And they are applying a deep learning approaches. So this is all about artificial intelligence. So we'll just do the most basic of basic walkthrough of these things. You could likely spend maybe a year on each one of these individual bullet points alone, talking about it. So please, this is the simplest. So first we have machine learning, and that's all these are big words, hot words. So what is machine learning? It's a combination of algorithms and statistics. So what do? They find patterns, and they're using big data. So this data could be images, numbers, likes, clicks, searches. Maybe some of you are participating in that right now, liking something on Facebook or Twittering, liking a tweet or something like that. loving a tweet, you're participating in this machine learning data collection right now, and they're tailoring it to you, what you do, so that they can go, oh, well, you like that. We think based on our algorithms, we think you'll like these things also. So imagine that with images and diseases. When we identify something, then we can say, oh, here are some other images. See if you can find something like that. So a subset of machine learning is deep learning. So deep learning is machine learning with the addition of these things called deep neural networks. We'll talk about that in a second. So deep learning is sort of like machine learning on steroids. It amplifies the machine learning, and so you can identify even smaller patterns. And you can use many, many layers. So as we go back to this layer point, all these pieces of data, all of these different data that we collect, maybe not even imagery data, but we can collect all these other things, and these are all layers. And we can use that and input them so that they can make predictions, find patterns and make predictions. So neural networks, part of deep learning. So they're using inputs. We have some sort of input. We have these hidden layers, which is where these nodes take place and they make a decision with weights. And then from those weights comes an output. We can train these neural networks. And so here's an example of an artificial neural network, which was done for corn and soybean yield prediction in 2005. So we have the basic flow, we have our input layers moving into hidden layers, and then we have our output, and then predicted, and then what we observe. So we can gauge how well they did. And they show this here. So these are yields. And so the figure on the top is their neural network for yield prediction, and this is of soybeans. And so we can see there R squared is, it agrees with the actual, which they collected much better than the models they used below. And they also reduce the error as well. As you can see, the RMSE is lower in their neural network than it is in the other. So there is certainly, not just across identifying disease, but this is predicting yield potential for machine learning neural networks to enhance our understanding of what's going on, not only for identifying disease, but for counting nematode eggs, but predicting yield. And so training is important. So there are different types of learning. We have three main paradigms, and there may be more, and some may, and I'm sure we could discuss or talk about why some aren't included, and you may very well be right. They should be, such as semi-supervised. But here are three that I find. So supervised learning. So as you can guess from the name, we take our images and a professional looks at those images. Say it's those images of frog eye leaf spot on soybean. And we circle those lesions, we identify those lesions as being frog eye leaf spot. So we're pre-labeling our data sets. And so that could certainly take a great deal of time, and it does. And we need many images for the training. So that means all those images need to be labeled accurately. So another is unsupervised, where they get zero. You don't label anything. It's just these raw data, and then it's sort of like saying, here's all these things, you go through them as you will, you find the patterns, you tell me what you think, I will give you nothing. Then we have reinforcement, which will focus on supervised and unsupervised primarily, but reinforcement is a more of a closed loop where we maximize the reward structure. So sort of like you penalize or reward based on the output, and if it's right or incorrect. And so let's talk a little more about unsupervised learning. So unsupervised learning is previously it was sort of thought of as a black box when you did unsupervised learning or used unsupervised machine learning, because you would give it your data and then it would make some decisions and it would come to a prediction, though how it came to that prediction was unknown. Therefore, the black box. So that's a bit disconcerting when you say, well, that's amazing that you ended up identifying this correctly 99% of the time, but how did you arrive at that prediction? And how did you do so well? So trusting something that you can't understand is, and then relying on it and relying on that furthermore is difficult, especially for researchers who like to know why and what and how this is happening. And so I really like this image, and it's looking at these unsupervised models, and perhaps you're not doing the right thing. So we can look inside the black box, and let's look at this first one. So first, just a researcher doing a task, looks like they're looking at different leaves on plates suspended in maybe water or something. And they're looking at reflectance, and the first one has no disease. The next plate, we don't know what it is. Maybe it's just a piece of green paper. The third has disease, so we have a red check mark, and then the next one, the fourth up there, has disease also, so it has a check mark. And then we can see below those green leaves that apparently we're identifying these spots on the leaves and how they're reflecting light, as we see to the right, the reflectance. So that took a long time. And so our friends go, oh, why don't you use some deep learning? Well, we don't know what deep learning is, but we'll try it because seems like everyone's doing it. That's what we want to do too. So we plug it in, we give it our deep learning, and well, it does fantastic. A lot better than a lot quicker than I did. But that's not enough. How did you arrive? How did you do so well? I'd like to know. And maybe now it's been shown that we're able to look into the decisions and what layers and what the deep learning model is using to come to a decision, what they rely on most. And we do that and we look into it, and it turns out that, looks like you're making the decision based on a pattern you see of the reflections off the plate or the surrounding water in the plates. Well, I don't really like that. That doesn't make much sense. I don't think you could use that in other instances. And so this was referred to as clever Hans strategies, because clever Hans was a horse who could supposedly count, but he couldn't count. He was just drawing cues off of people. So he was arriving at the correct answer, though not by appropriate means. And so the researchers here, they are moving towards modifying how the deep learning model chooses what to look at and what to go off of. And so before that, this is more applicable to soybean diseases. So this is a high impact paper from researchers out of some from ISU also. So it's an explainable deep machine vision framework for plant stress phenotyping. So here they had over 25,000 images for training their models. That is a great deal of images. So I always thought that would be easy. I'd just send my sister out. She could take pictures as much as you want. She likes taking pictures of herself, but that is a lot of images. And not only do you just take the images, you need to, as we talked about before, label these images accurately. And if you're training your model, unless you're doing unsupervised. So here in this paper, they were able to accurately identify from their model bacterial blight, bacterial pustules, septoria, SDS, frog eye, as well as some abiotic stresses, herbicide injury, potassium deficiency, and IDC. And they were also to take, as we spoke of before, take a peek at what's inside that black box. So why and how are you arriving at these accurately predicted identifications of disease? And this is very important because this can be applied to high throughput phenotype. So when you remove the person and you just have images, you can go through much more easily. So here's an image from their paper. And what they saw, they had, we can see the diseases on the top, the original image, which as what we would see if we were to walk out into a field. And then the prediction explanation output is what they feel is what the model is relying on. And then on the bottom, they have their marked image of the diseases. So they got an idea of where and what the model was relying on to make these predictions. And so what about the impact of disease? We know it causes a large dollar loss, and we can identify it. Researchers are identifying them with machine learning techniques accurately. So what's that mean for yield though? So common, researchers, we like the area under the disease progress curve. So we look at disease usually from the beginning of an epidemic to the end, and we rate for severity, and then we plot this severity across time. And we do this for different cultivars, phenotypes, and we do these. So here's cultivar R, we'll say that's cultivar resistant. And then we have our cultivar S is susceptible, and you can see severity wise that cultivar R was did not have very high levels of severity across this period, though S certainly did. So that's why we're calling it susceptible. And one of the things we look at, the area under this curve, we derive that. And so for R it's 200, and for S we'll say it's maybe 6. And we look and we say, okay, well, likely 6 will have a larger yield loss. It will be associated with a higher yield loss, or S is more susceptible, which it certainly appears to be. But this is not the best. So why is that? And it's because disease severity is not directly related to yield. But radiation use efficiency is, and that's RUE. So RUE is the plant's ability to convert photosynthetically active radiation into biomass. Biomass, where do we get yield? It's straight, it's a measurement of how well photosynthesis is performing, photosynthetic performance. And can you imagine what disease does on a leaf? Not only does it reduce the green leaf area, but it could reduce the photosynthetic capabilities of surrounding tissue also. So this is unreasonable on a small plot scale, and not to mention perhaps even for anything other than that on a farmer scale. But for doing many phenotypes, consider a commercial operation where you have thousands of lines, individual rows of all these genotypes. Can't be done. And we'll talk about why and what's needed to do it. And so still, there's not a uniform procedure for RUE estimation across crops. So this has been one of the factors attributing to some of the variation that we see in literature and for maybe individual crops in general, is that one group may use this way, another may use this way. So it's been proposed that we shift towards only considering the green leaf area when estimating RUE. And so we have our RUE green, which stands for the radiation use efficiency of only the green leaf area, is equal to the dry mass over the fraction of the absorbed photosynthetically active radiation of the green leaf area multiplied by the incoming PAR, photosynthetically active radiation that's coming in. So that there is the simplest part of RUE, that little bit, but it's not simple at all. It's difficult to collect, estimate, and everything in between. So it has three main parts. So first we'll look at dry mass. So we need to look at the above ground mass that's produced. We don't look at the roots. That'd be very difficult to do, to estimate. But perhaps in the future that could be included. Second, we have the leaf area index. So the leaf area index this is the primary border of energy and mass exchange. This is where everything happens. Photosynthesis, the most important chemical process on Earth. That's where everything is happening. And then when we consider foliar disease, well, it certainly isn't helping. Removing green leaf area does not help with yield at the end. And then we have our APAR, which stands for absorbed photosynthetically active radiation. So our green leaf area, our leaf area index, it is absorbing PAR to build this mass. And so that's RUE. So we need those parameters to estimate RUE. And so we'll look at APAR real quick because it has a few other components. So we need to know how much photosynthetically active radiation is coming in, how much is going out, which is reflectance, and then the fractional cover. But let's not forget about the disease severity. We need to remove the disease severity from our calculations, and so that's the green leaf area. So we need to remove our estimated green leaf area from the severity from the green leaf area. And so here's an example of one of the projects that I worked on. And so that was estimating RUE for soybean, partially from UAV imagery, consumer grade unmodified UAV imagery. And so the top is not the images we used. Those are just images of the whole field in general, so you can have an idea of how it progresses. But we did 14 sampling dates, and we calibrated this imagery using reflectance panels. And this bottom picture right there that we saw before with the 101 plot marker, that's only one of about 670 images taken at 30 feet above ground, stitched together to form a high resolution orthomosaic with sub-centimeter resolution, and those are what we used. And so we accurately predicted or estimated, I can't say accurately, but our predictions of RUE fell within the range of published values. So we can use consumer grade unmodified imagery to help move towards complete estimation of RUE. And so it's important that you note that it's consumer grade imagery. Those are what farmers are flying around or most easily to get. So we can apply this using those. And so this is just a step towards the absolute remote sensing of RUE. And so what do I mean by that? So what I did, I collected destructively some of the portions I needed for RUE estimation. What's next is to show that we can do it all through imagery or remotely. And of course, if you think of applying this in any other way, you cannot do destructive sampling for components on a large scale. Well, why is that? Well, because sometimes your plots are one row. And in general, it's just a terrible amount of work. And that's why I noted before it's unreasonable. But we can, it's been shown in literature before that we can derive LAI and biomass from imagery and predict those things. And then the last part of everything is to incorporate these trained algorithms so we can identify accurately diseases and the severity, so we can incorporate that into our RUE estimation to get a better guess, not a better guess, better estimation of what's going on. And so I'll finish with where we started. So the future of foliar disease identification, quantification, and impact determination is promising. And it's collaborative. So we need collaboration across plant pathology, computer scientists, engineers, so that we can move into this new era of data collection and disease determination and impact determination. So it's also accurate and precise. We're trying to remove that human error that comes with me when I go into a field. I get hungry, very hungry when I'm out there rating, and it most likely won't impact what I do. And that's just a fact. Well, we can help reduce some of that error. And this is, it's big data. There's a lot of data, a lot of layers going into it. And perhaps maybe someone will have to come up with another term to describe the sheer amount of data that's going to be collected. But thankfully, the computational abilities that we have access to now are going to be able to handle that. And this will be real time. So all of these tools that are being created, not only just to identify disease and assess the severity and RUE, but this will be real time. These algorithms and these models will be implemented on the UAVs as they fly, and it will be automatic. No more waiting to send something into somewhere to see what it is, or it will be instant. Not only that, but it will be applicable across scales. So the farmer to the researcher. And that's important. So, and with that, I'll say thank you. I know I'll be taking, hopefully you have some questions. And if we don't get to all the questions, please, this is my email that you can reach out to me with. And I'll hopefully get back to you. So thank you very much.
All right, Xavier, thank you very much. We appreciate you for that presentation. And folks, please consider submitting your questions now. I do have some coming in, but if you want those answered in the next 10 minutes before we wrap up this session, go ahead and submit them. All right, Xavier, so just some questions to kick it off, and then we'll keep moving until we run out of time. First of all, this individual says the AI that you're talking about today is very interesting. It helps to manage pesticides, do other things. Can this technology be used for natural systems like enhanced nutrient and probiotic programs to improve yields and reduce yield losses? And then from there, I'm going to ask you another question. So take that one first. Can this technology be used for natural systems like enhanced nutrient and probiotic programs to improve yields and reduce yield losses?
Well, so I don't have any experience with that, but I believe certainly there is examples of it being used for these other types of systems. And it's, as I didn't go too far outside of disease identification and counting eggs, but using AI and machine learning to do what it's supposed to do, identify patterns. These patterns certainly could be for what you asked. So I think so.
All right, great. All right, thanks for that. Next one. This individual says you spoke about how computers could be used to measure or determine the number of nematodes. Is there also a way to measure root mass using, AI technologies, phenotyping resources, any of those? I think this individual is driving at. You touched briefly on roots. Maybe talk a little bit more about that.
So when I talked about roots concerning what we can look for dry mass for the RUE estimation. As far as determining root mass from AI, I do not know. I haven't looked. I haven't seen any literature that I've read, but that's a very small amount. I would encourage you to look. I haven't seen anything personally, but
Sure.
I also haven't looked very hard for it specifically either.
Okay. Yeah, no problem. I guess I would note that there are some root phenotyping programs funded out there by the Department of Energy now where they are looking at roots. They're trying to estimate root mass and a lot of other particular traits of roots using, I guess you could say, non-invasive technologies. So it's coming along, but there's a long way to go on that technology. It's very difficult research, and we'll see how it comes about with implementation and application. All right, a question that I think will be very interesting to hear the answer to is, Xavier, what do you do about multiple diseases and measuring them and identifying them at one time using UAVs and some of the other techniques, data analysis techniques you mentioned today? Multiple diseases one time.
Sure. So multiple diseases are hard, especially when it's a person in the field. When you come across a leaf with lots of issues and segregating those and just trying to rate one. But then when you go from human to machine learning that's hopefully trained and able to identify the different diseases that you're seeing on these leaves, I would expect that the error is much less. And then you touched on a point that I perhaps should have addressed. or I thought someone might ask, and I think you touched on it, the disease from the UAVs. So when we talk about just getting an idea of diseases from UAVs, we have to remember that our UAV imagery is only collecting what we can see from looking down. So what about those diseases that are below the canopy? We'll be missing those, so we may be under our severity ratings may be lower. So that's where not only will UAVs be a big part, which they're a part now, but a bigger part. Another portion will be perhaps rovers on the ground that are able to go through the field and get a better look at things below the canopy. So this has been done, a recently published paper using these robots that go into the field, and they scanned corn plants and gathered different parameters on corn. And so I think it'll be a combination of both UAVs and those other, these rovers, robots, all at once collecting data so that you can get a better idea, because not only not on the same leaf you may have other diseases, but lower in the canopy you have other diseases that you may not see. And so it's a combination of these things to get a clear picture of what's there. So
Thank
hopefully.
you. Yeah, that's good. No, those are fantastic points. Appreciate those comments. I'm going to keep rolling here. We got just time for a couple more questions. We have more than that, but we got to keep going here before the end of the hour. So next, which indices were you particularly using to train your models and the research that you were doing?
So I didn't train models specifically in my research. I was just estimating RUE. The models that I was speaking of, or the AI models I was speaking of, was sort of the last component of moving RUE into this complete remotely estimated parameter, to include disease estimation. But at least in my research, I used NDVI and the excess green index, particularly to discriminate between green leaf area and background noise.
Okay. All right, thank you. The next question is a great one, and I think a lot of people have thought about it, but we're not sure we got an answer on it yet. How do other abiotic stressors present at the time of disease identification and measurement impact a model's ability to differentiate between disease types and assess disease severity? For example, what are the impacts of water stress and nitrogen stress on identifying and measuring disease severity?
Well, I think it will all go back to the model's ability and what the model was trained on. So certainly it would impact anybody just in general when you have multiple disorders showing up. Like we talked about multiple diseases, but when you throw in abiotic stresses, and many of them look similar to each other, perhaps determining what those are and delineating between them would certainly be more difficult. So I would stick with my first part. It all depends on the model's ability and what the model you're using has been trained for.
Okay. All right, very good. Appreciate that. All right, well, we are into our last minute here. And so before we finish, a few people that I want to thank. First of all, Xavier, thank you so much for coming today and doing this. Really appreciate you showcasing your research and all that you're doing, and also represent the Iowa State program, and really give us a lot of great information on drones, artificial intelligence, machine learning, etc. A lot of great stuff here, some cutting edge things going on. Thanks for doing that. I want to reach out to United Soybean Board and thank them. Again, their sponsorship brought this to you free of charge. Now, one note from the American Society of Agronomy, that's this. Sponsorship of this webinar from USB or any of our other sponsors for any of our webinars does not imply endorsement of a sponsor's views, products, technologies, or procedures by the American Society of Agronomy. But overall, thank you, United Soybean Board, for your sponsorship today. And folks, please go and check out the Tech Tool Shed, which I had up on the screen at an earlier time. It might be a great resource for you. With that, I want to thank everyone for coming today. Please be on the lookout for the recording and the slides. You will have access to those. Be on the lookout for future webinars. We have one coming up this Thursday that you might very well be interested in related to soil health. So with that, thank you, everyone. Have a great day. Take care. Bye-bye.